Talk about the sliding filament theory of muscle cell contraction. So at this point you are aware of overall muscle structure, the macroscopic structure, and you have zoomed into the microscopic level. Let me just review that with you real quick. I've got a little video that kind of will get us to this point, and then we'll talk about the details of how a muscle cell contracts. Okay, so let's just check this out real quick.
And move her hand to take notes. Your cardiac and smooth muscle tissues are involuntary. You do not consciously control their actions. Skeletal muscle works under voluntary control. Skeletal muscles are composed of bundles of muscle fibers. Muscle fibers are long cylindrical cells containing several nuclei. Muscles will contract or relax when they receive signals from the nervous system. A neuromuscular junction is the site of the signal exchange. This is where the synaptic bulb of an axon terminal and muscle fiber connect. Muscle fibers are composed of many myofibrils. A myofibril contains contractile units called sarcomeres. Sarcomeres run adjacent to one another down the length of the myofibril. Each sarcomere consists of alternating thick and thin protein filaments, giving skeletal muscle its striated appearance. The muscle contracts when these filaments slide past each other. The thick filaments are myosin, which are anchored at the center of the sarcomere, called the M line. The thin filaments are composed of the protein actin, which are anchored to the Z lines on the outer edges of the sarcomere. Because the actin filaments are anchored to the Z lines, the sarcomere shortens from both sides when actin filaments slide along the myosin filaments. Although the
Okay, let's take it from here, all right? So we are at that level. That's where we are. So you can kind of review that at your own pace if you need to. So here we are. We've got the actin filament and the myosin filament and then another actin filament. Okay, so it's a repeating series of units. I always remember the actin is the thin filament. I think of this. Let me bring this over. That actors are too thin. All right, so that's my way of remembering that. Actually, I shall just bring it down here and leave it there. And there is the actin, as they call it, a myofilament. We'll probably just call it actin most of the time. And then the thicker one, as we said, is the myosin. Okay, and now, what about these little purple guys? All right, so a lot of times in life sciences, purple is used to represent proteins. Okay, we're just going to overgeneralize here and just call these proteins regulatory proteins. You're going to hear in another video clip, I'm going to show you that there's specific names for these regulatory proteins. We have bigger and better fish to fry, and we're just not going to spend a lot of time on what those proteins names are, just to spare you a little bit, but just recognize for now that they are in place and they are preventing the myosin, and then more specifically what's called the myosin head, from attaching to the actin. All right, and so it's this attachment, it's the attachment of this myosin head to the actin that is going to initiate the muscle cell contraction. Okay, and so what you can think of, is you can think of people rowing crew. Okay, so this would be the myosin head, and in the water would be like the actin. So you got to get that myosin into the water, and then you've got to move it. Okay, in this case, the myosin is like the oar, or the blade of the oar, and it's got to move. And so once that movement happens, that you're going to get movement of the muscle cell itself. Okay, so let's let's just stick with this for now, and I'll come back to the rowing action. But I just wanted to plant that seed in your head. Okay, so now how are we going to get this protein out of the way? Okay, well, protein is a crazy complex structure. All right, it has all kinds of elements to it. I'm going to just show you a real brief segment of this video just to show you some of the complexity of a protein. I think that will help as we try to think about these almost as basically like little machines. All right, so let me grab this real quick, and I want to streamline it for you. So let's bring this in right here. Kind of think of a protein backbone. We've got carbon, hydrogen, nitrogen, all that good stuff is in here. You know those things. All right, let's check out a little bit of this just so you can see how dynamic a protein is, and so you can sort of imagine, hopefully, how it could change shape.
In a carboxyl group, a water molecule is released each time a bond is formed. Specific amino acid sequences give proteins their distinct shapes and chemical characteristics. These protein chains often fold into two types of secondary structures stabilized by hydrogen bond. A protein chain can fold into a rigid alpha helix, forming regular patterns of hydrogen bonds between the backbone atoms of nearby amino acids. Backbone atoms of the chain can interact side by side to form...
Okay, actually that's pretty good right there. Just seeing that folding action. You saw hydrogen bonds, you've heard of that before. So just the idea that this is a complex shape, all right, and it just gets more and more complex, okay? So you've got a structure like this, and then you can even have it as complicated as that. Okay, this is hemoglobin. So anyway, proteins are crazy. They can change shape. They make everything in the body work, essentially. It's just a very abundant molecule. Okay. So how are we going to get these guys out of the way? All right, so enter one of our most talked about ions. Get that out of there. And that would be the one that we always talk about when we talk about bones. And we talked about osteoporosis. And we said if you don't have enough of this in your diet, your body will go to the bones and mine the bones, so to speak, use those osteoclasts and osteoblasts to get in there and mine those bones out. And so you've already probably figured out what this is going to be. I'm not with the American Dairy Farmers Association, so I don't have to say milk, but I could say milk is a good source of calcium. So anyway, it's calcium, all right? We got to have the calcium. And what the calcium is going to do, and what's being shown in this diagram, is that the calcium is attaching to that protein. And so I think now you can probably imagine that the presence of something with a charge could reconfigure the shape of that protein. I mean, it's crazy complicated. It's at a cellular level. This is cutting edge stuff. This is a theory that's been worked on for 50 to 60 years. We're still, working on it. So, but this is what we believe happens, how it goes down. So the calcium attached to the protein, and what they've shown here is basically that it's revealed a myosin binding site. And so the idea is now that this myosin is attached to a myosin binding site. There's going to be a new vacancy available here in a moment for the myosin to attach to. This will be a binding site. This myosin head is already attached to a binding site. So essentially, the presence of the calcium allowed these regulatory proteins to shift out of the way. All right, so we moved them. All right. Okay, great. So now we've attached ourselves. The myosin is attached to the actin. Okay, good. Now, actually, I want to keep this screen here like that. So now, notice the difference. Compare the difference between this myosin head and this myosin head. All right? What's different about them? They're in a different position, a different shape, however you want to say it. This one is aimed off to the right. And this one we would say is cocked back to the left. Okay, and so actually the action of going from here to here is known as a power stroke. All right, and so when the contraction happens, all of these myosin heads, and we're just showing 4 here, there's going to be a gazillion of them in one filament, and a gazillion filaments in one cell, and a gazillion cells making up one muscle. So obviously we want to oversimplify this a little bit, but at this level right here, I think we can see that between here and here, we've changed shapes. And more importantly, we can tell that the gap between these actin filaments on either side has narrowed compared to up here. Okay, so when these myosin heads flexed in or cocked in and made that power stroke, they brought the actin with them because the actin, as you remember, is attached to what would be a Z line over here on both sides. And if we were able to show the Z lines on both sides, this whole thing would be considered a sarcomere, this little functional unit within a myofilament. All right, so that happens over and over again within the filament, bringing in all of these actin filaments and therefore shortening the muscle. That's what muscles do. All right, they contract, they get shorter, they get smaller, and they relax and they go back to the way they were. All right, and so the result is we've got to look at a little rowing action just for fun. Somebody had a good time putting together the best rowing video ever. I don't know how I could judge how good a rowing video is, but here it is. So I'm going to kind of use this as an example. All right, there's those myosin heads hitting the actin. Boom, boom. Yes, nicely done. All right. Okay, and the boat's moving. The actin's moving. That is my favorite part. These guys are all buff, standing there, and then there's this dude chilling in the back. All right. So anyway, think of the rowing of the boat. Make sure you've got the right vocabulary in place. I think you'll be in good shape figuring out the sliding filament theory. Thank you and enjoy.